Welcome to the lesson on bacteriophages. This is one of my favorite topics. And before we start the lesson, I just wanted to show you this picture from my family album. There's me and my daughter Paula when she was little. She's grown up and married now, so this picture is probably from the late 90s. And it features a 6-foot tall bacteriophage. And the reason I made this was that it was an ad for a smaller exhibit where kids put together little models. But I just thought I absolutely had to have this giant phage, and so it's just made of cardboard and 2x4s and junk I had in the garage, a little bit of silvery fabric from the fabric store. And it lived in my garage for many years, and then sadly there was a little bit too much wear and tear and I had to disassemble it. But I have very fond memories of my 6-foot phage. And this is here to be an advertisement for the activity. At the end of this lesson, you can download the pattern to make this really nice little tabletop size model. Now make sure your pencil's nice and sharp for this lesson. I usually don't worry so much about sharp pencils, but we're going to draw some very tiny exact things in these spaces. I apologize ahead of time for trying to get so much into one lesson, but I really wanted to get it all on one page. So make sure your pencil is sharp and you have a sharpener handy to keep it sharp. And we're going to start with drawing a T4. But before we do, here is an electron micrograph showing you some of the very first pictures we ever had of phages. This is a bacteria. So you can see these guys are about 200 nanometers tall. Remember, a bacteria is about 1,000 nanometers, which is a micron. So these guys are much smaller than a bacteria. And you can hardly see their little tail fibers. We're going to draw some tail fibers that are almost invisible in this picture. So our T4 is going to have an icosahedral head, like most other viruses do. And I've given you some starting dots so that you don't have to worry too much about drawing a perfect hexagon, because I don't want to waste time on that. So play connect the dots for your outline. Down to that collar looking thing, and it actually is going to be called a collar. And then you remember how to make it into a icosahedron. Keep your lines very light because we're going to erase some of them. We're going to erase this side in just a minute. So just very lightly, like that, and make your top triangles like that. And remember, next we make two coming down like this so that they form a third in the middle. That sort of. Again, keep your lines really light. These are just guidelines. We're going to erase them in a minute. And then we have a line coming up like that, and then the two on the bottom like that. Now draw a line right down the middle and erase the ones on the right side. This is where we're going to see the interior. So on the left, we're going to have an exterior view, so you can kind of shore those up a little bit. You can even shade just real lightly on these, make them look like they're kind of on the underside. And then we're going to draw a line like this just inside here. So we're going to see like the thickness of the capsid. Let's go ahead and label this top part. It's called the capsid. It's a word we've seen before. And let's put in parentheses head, because this is often just called the head. A phage has a head and a tail. There's no body or arms or legs, just a head and a tail. So everything from here up is the head. And then you can leave it like this if you don't like to shade, but if you do like to shade, you can make this look sort of hollow by starting out like that, making it dark along there, and then just get lighter very gradually get a little bit lighter, a little bit lighter, a little bit lighter, a little bit lighter. Use your finger to smooth it out. If it looks like a stripe, that means that your transition between light and dark is not gradual enough. Okay, and then I like to shade a little bit around here, make this a little bit darker, like that. Now, having your pencil sharp for this is not going to be helpful, unfortunately. You can kind of tip it to the side a little bit. Okay, don't make it too dark though, because we're going to be drawing some DNA on top of it. And then inside we have the DNA. And it has a lot of DNA. It's full, packed full of DNA. So let's make a bunch of squiggles. It's packed full of this stuff. And we'll label it DNA, deoxyribonucleic acid, which contains all the instructions. What would the instructions be for? How to build a T4. So let's just say the DNA codes, it's like a secret code, codes for how to make 289 viral proteins. So it takes 289, as far as they know now, tiny individual proteins to make up the T4. And then if you want to put a line here, I want to say something else about the DNA and how big it is. So you know DNA looks like a twisted ladder. So there are 169,000 rungs on this ladder. And here's a reminder picture. Each rung is made of a pair, 1, 2, is a pair of bases. And the bases are little molecules. Remember, they're cytosine, guanine, adenine, thymine. They're made of atoms. So it doesn't really look like a bar. It looks like a group of atoms, but it's easier to draw a bar. So this is called a base pair because it's a pair of bases. But informally, we can call it a rung. So the DNA of the T4 has 169,000 rungs. And then we can put in parentheses base pairs. And this is very important information for viruses. They keep track of exactly how many rungs are all on their DNA. You can find charts on the internet comparing all the different viruses and how long their DNA is. Now we're going to draw this little collar piece next. Right under here, we want to draw just sort of a same kind of a curved shape like this, just a little bigger than that one, and we can label it collar. Make sure you keep the letters small because we're going to be writing some things down here. And then we're going to be making the tail sheath. See these dots down here? That's where we're headed. So come right under here, sort of like where these lines are. Go like this and go right down to here. And this one to here. And I'm going to make a curvy line like that. And then sometimes they add some texture lines like this. You don't have to. They're actually kind of lumpy and not real super smooth like this, but there's good. Looks nice anyways. Shade it a little bit. And this is called the tail sheath. So everything from here down is part of the tail. This part is called the sheath. Tail sheath, S-H-E-A-T-H, just like the sheath that covers a sword. It's kind of like a covering thing. And then right under here, the top of the sheath, right in here, we'll make some things sticking out like this. And if it looks like whiskers, that's exactly what they're called. Whiskers. They think the whiskers sense the environment just like a dog or a cat's whiskers would. And not all phages have whiskers, so it can't be absolutely essential to all phages. But with the ones that do have them, they think that they sense when is a good time to put out these long tail fibers we're going to draw down here. So if it's not a good time, the whiskers will sense that and they'll keep the tail fibers retracted close to the sheath. So let's go down here and draw in the tail fibers, but they're connected to a little base plate we have to draw first. So just draw a thing like this and kind of up and down a little bit, like that. And this is the base plate. I think they make it all one word. Here's a better picture of the base plate, kind of a close-up. You can see how the tail fibers are coming out of it. And there's the sheath, the whiskers, and the capsid with a beautiful hexagonal pattern all over it. And then coming out from the base plate, we have those things that look like legs. And I'm going to make mine like this. I'm going to go stay kind of close here, make a sharp angle up and down for the first one. I'm going to have 3 on either side. And then come out like this for the second, up. And kind of make it a wider angle down to here. And then third one out like that. They don't have to look just like this. You can, whatever you draw will be fine. Up and down, and down. Now they call these the long tail fibers, for obvious reasons. So where are the short tail fibers? Well, believe it or not, these things right here, they actually call tail fibers. I know they don't look like it, but so let's add base plate with short tail fibers. So each of these little things and these have the ability to attach to a bacteria cell wall. So let's put little dots at the end of here like this. Make that one a little bit longer there. And let's label these. We'll call them receptors. That's kind of a general word for a little attachment point thingy. Receptors, R-E-C-E-P-T, receptors that will stick, or we could say lock on would be even better. Receptors that will lock on to surface parts of then we could say bacteria, or we'll go ahead and say which bacteria it's going to stick to, and that's one called E. coli. Capital E period C-O-L-I. And we'll take a look at E. coli in just a minute. So right here is going to fit into a little part in the surface of the bacteria. And in this space right here, we got some nice empty space. Let's write how tall this is. This is 200 nanometers tall. And if we look back at lesson 2A, where we drew all the viruses in comparison to each other, we'll see that it would be about the same size as herpes. And in fact, if you want to go back at some point and you want to draw a little bacteriophage in there, that is where the phage would fit on our chart. Phage 200 nanometers. You don't have to do that now, you can just do that later. And then we're going to go over here and look at some other shapes that these might take. So the T4 belongs to a group of phages called the tailed phages. And not all phages have tails. So I thought we could look at, let's zoom back out and look at this picture where somebody has drawn some various morphologies. That's what they call shape, remember, morphology. So here's the one that looks the most like what we're drawing. But you can see some of them have little tiny tail things and no fibers. Some have little spiky proteins coming off. Some of them have curved things and little straight tail fibers. I keep wanting to call them legs, they're not legs. This guy's got a kind of more of a spherical shape to its capsid. You can still see the icosahedron under here, but it looks spherical. And then it's got tiny little fibers. And this guy's got thicker one, and this one looks like, you see how it's like holding on to something? It's got a curlicue fiber going off, and it looks like it's holding on to this thing, whatever that's supposed to be. Let's make this first one look like this guy. Should have shown you this before. Okay, so let's round it out a little bit. And then just put some little spikes like that coming off. Maybe one in the middle, like that. We don't have to know its name, just some of them look like that. And then this one, let's make it look like this guy here, one of these guys. Okay, so we're going to give it a little tiny tail like that, and then maybe add some spiky things going off like that. And then this one, let's make it kind of like our T4, but with a real short tail sheath. And then let's go ahead and give it some fibers. So the difference between this one and our T4 will be that this one won't contract. We're going to find out that our T4 kind of scrunches down like that. And this one's already scrunched down, so it can't do that. And this one, let's give it a long, thin tail sheath, sort of like ours, but then tiny little, let's give it shorter fibers like that. And let's see, down here, let's do something at the bottom first. Some of them look like this, so I can hardly get it all, and they're just like little tubes with a base plate. Kind of reminds me of the TMV tobacco mosaic virus. So let's put one of those down here, just a long cylinder with kind of a little base platey thing at the end. And then this guy, let's do kind of a curvy, like this. And then straight little fibers. And then this one, let's give it a curvy tail sheath, and then give it a curlicue tail fiber like that. Now we're going to come over to this blank oval here, and we're going to turn it into the type of bacteria that T4 infects. Now remember, bacteriophages, phage means eat, So they're bacteria eaters. They don't touch any other type of cell. They can't because their little receptors on their tail fibers will only fit into things that are on the outside of this type of bacteria. So the type of bacteria that this will infect is called Escherichia coli. Now Escherichia is a long word, and so nobody wants to say it, so it's known as E. coli. And you may have heard of E. coli. Sometimes people talk about it in terms of food poisoning. And that's true. There is a strain you can get that causes sickness, but mostly E. coli is a harmless bacteria that just lives in us all the time. It's part of our microbiome. And it's just if it gets out of proportion and there's too many of them, that it gets to be a problem. As long as the population of E. coli stays down where it's supposed to, there's no problem. And it actually helps you by producing a vitamin that you need. And we're going to start by giving it the feature number 1, the flagellum. And this is kind of looks like a tail. This is the most exciting part we'll draw. So just kind of give it, we'll loop it around and make it stay on the page there. Sometimes the E. coli will have more than one, but let's just give it one. And let's label it, point to it, and put a number 1. So that's a big difference between the bacteria and the virus. The bacteria can move. It can use this little tail and whip it around and kind of sort of swim. So number 2 is the bacterial nucleoid made of DNA. So it has a clump of DNA in here. So just draw a little tangly mess. Now sometimes the DNA in a bacteria is actually a circle. If you were to untangle this and stretch it out, you'd find that it was actually circular. You can imagine taking a circle of string and just clumping it together, and it's going to look like that. Sometimes bacteria will have linear, but oftentimes they have circular. So let's label this as number 2. And then for 3, we're going to draw a line like this around that line. And this is going to represent a second membrane. So the whole thing, all the outer coating, is called the envelope. And different bacteria will have different structures in their envelope. This one has 2 membranes. All cells have at least one membrane. That's required. Plant cells, animal cells, bacterial cells, they have this inner membrane. But this guy has an extra one. And we'll take a close-up look at it in the next little picture. So let's put a 3 on this. And then also let's add some little tiny little hairs. You'll see what these are in the next drawing. These are little, actually strings of sugars, oddly enough. You know, glucose is is the individual sugar unit. And if you string them together, you get starches and things. But sugars don't just get digested for energy. The sugars can be used to build things. And in this case, there's little strings of sugars attached to some fats. We call them lipopolysaccharides. And they hang off here, and they're part of the bacteria's defense systems. And then make some really light lines kind of crisscrossing everywhere. They did not used to think that bacteria had a cytoskeleton like plant and animal cells do, but now they know they do have these thin protein cables that stretch across, and they give it kind of a framework structure. They help it to stay round and not squish flat. And so this is called the cytoskeleton, and that means cell skeleton. So let's, I don't know, you can point to it. These, well, somewhere. The stuff. Number 4. And then this is the most important part of the drawing, probably. The E. coli has something that the virus doesn't. and that is ribosomes. So let's make some little round dots and label them ribosomes. Number 5. Ribosomes are little factories. They're little manufacturing sites where proteins are made. And they have the ability to, by way of a messenger molecule, read this DNA. So the DNA has instructions that tell how to build proteins, which are the parts of which things are made. And these guys are the actual factories that make the proteins. And the virus doesn't have it. The virus has instructions, but no factories to make the stuff. And then let's add some little tiny dots. This is where you need a really sharp pencil. Tiny, tiny dots, even smaller than the ribosomes, much smaller than a ribosome, are some of the little things that the ribosomes have made. In fact, I think all these have been manufactured by the ribosomes, and they are like little task robots. Each one of these things can do something. So that would be number 6. Point to one of those little tiny dots. We call them enzymes, but I like to think of an enzyme as a task bot. And each enzyme can do one thing. So if you have a lot of jobs that need to be done, you need a lot of different kinds of enzymes. And then we'll add one last thing, because you'll see it in some pictures and you'll say, why didn't our picture have that? So let's put it in. These are little areas. They're called inclusions. Number 7. And they are places where viruses are being made. Viruses aren't made randomly all over the cell. They're made in little centers so that all the parts are all together. And let's add, just for scale, let's add some tiny little T4s like this on the outside of our E. coli. They might even be smaller than this, but this is about how small we can draw. But that's actually not too bad compared to this picture. It shows some T4s hanging on. That's pretty good. And then we're going to come down here and briefly mention this before we go to this close-up, because it's like this picture here is a close-up of what's going on here. So we could zoom in on what the T4 is doing. This is what we would see. So let's draw the outside of the E. coli right here. Let's make these little 2 membranes. So down here would be inside E. coli. And then we'll make our T4 pretty big here. We'll make a hexagon. You don't have to make it look all perfect with the icosahedral. You just do a hexagon there. And then there's tail sheath, some tail fibers. Actually, I should put these little things on like we did. These are actually important in this drawing. Go ahead and put those on. So then whoops, forgot the whiskers there. The receptors on the tips of the tail fibers there are just the right shape to match part of these little sugar chains hanging off. So it will latch on, and it could possibly get shaken off at this point. It's kind of the first stage in hanging on. But then what it's going to do, so this will be the same one but later in time, draw the hexagon down here like this, so the tail fiber is going to be really short. So what it's done is go scrunch. So the legs are going to look like that. And that base plate is what's hanging on right here. So we'll see, the base plate attaches, and once that attaches, There's no shaking this guy off. And then it's going to inject its DNA. Can make a little DNA coming out. It's going to go and squirt the DNA into the E. coli. So let's take a close-up of right here. You don't have to do this, but if you want to make a circle, this is what we're going to look at. Right here, we're going to see the base plate and a little bit of the membrane, and we're going to see the DNA right before it comes out, but we're going to take a close-up of that. So here we have the base plate, and I'm going to use a little color to help me keep track of what's what. I'm going to make my base plate blue, I think. And then you see how like it has a particular shape there that's locking into a part of the bacteria. So that little shape around there, okay, that would be, that's something different. This is part of the bacteria. I'll make this pink so it'll show up. That's part of the bacteria's wall. So see how it's locking in? Okay, so let's see, I'm going to make the like sheath, maybe a different kind of blue. You can make it any color. You don't have to follow my color scheme. I'll make it kind of look like that. So that's the bottom of the sheath, and this is kind of like the inside of the sheath. Maybe that's why they call this the sheath. There's this kind of inner part to it, like that. It's amazing we even know this structure. When I was in your age, they had no idea of this, what this looked like. This is all in the last 10 to 15 years that they've found out what this looks like. So let's go ahead and label this. I have number 1 down here is the base plate. So we can point to this as number 1. And let's see, I have DNA as number 2. So the DNA would be inside here. Can make a squiggle going that way and then the opposite way, and then make some little rungs. And there's the DNA waiting to come out. Number 2. And then this thing right here, I'm going to make it red so it stands out. This is a strong piece of protein that acts as a needle. I'm going to make number 3 is our protein needle. And you need something sharp to poke a hole in these membranes. However, there's a layer right here that is, this is number 4. So see that middle layer? I'm just going to label it number 4 on the edge like that. This is made of proteins and sugars. The little ovals are the sugars, and then there's little lines going through. Those are the proteins. And so this is like a little fence that is held together pretty tightly. And to get through here, you need a pair of clippers that can clip the fence. It's like you have a chain link fence. You need a pair of wire cutters to go in and clip, 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 you know, cut all the little wires so you can open the chain link fence. And so the wire cutters are these little things right here. They're called lysozymes. So if you point to those blobs, those are 5. And I'm going to make mine, let's see, a new color I haven't used. I don't know, maybe green or something. And I'll show you a picture of that in just a minute. Let's finish labeling here. Number 6, the outer membrane, be right here. And number 7 down here, the inner membrane. And if you want to add a little color, what I would use would be yellow, because just like a little bit like that. They are made of some molecules called lipids. Lipid means fat. And you see up here how it's already torn? Top there, the needle has already punctured that and torn open that membrane. The bottom one isn't torn open yet. And we can see some little proteins sitting in here. Membranes are full of little things sticking into them like that. You don't need to know what all they are, but know that membranes have little things sticking in them. And here's our tough layer. Here, right, that's that protein sugar. This is like the fence. And then let me show you a picture of the actual molecule, the lysozyme. This is the lysozyme molecule. It's an enzyme made of protein, so the string is made out of amino acids. Sometimes the string coils around, does a fancy shape like this called a helix. Other times it zigzags back and forth and makes kind of a sheet, which they show as a long flat arrow. And these things sticking down right here, they're like yellow with red tips. This is the active site. This is where the action happens, and the action that this molecule does is cutting peptidoglycan. So you could almost imagine that this is like a scissor machine, right? And then you could put the little peptidoglycans in there and go snip and cut them apart. Now the interesting thing is this enzyme is found not only in the T4 virus, but also in our own cells. You can find lysozyme in our tears and saliva and other body secretions, and this is part of our innate defense system against bacteria. So here's something that you have in common with a virus. Now before we leave this picture, I want to say one more thing about these little things sticking up here. Let's draw a tail fiber. It would probably be further out. It probably really wouldn't be in this picture, but let's just put like this, a tail fiber down here connecting to that, because I've mentioned that this is one of the things the tail fibers can grab onto. I think there's some other structures too. There's some texts that mention pores it can grab onto, but let's do this in our picture. Let's label this tail fiber. And over here, let's point to these and label them toxic. That means poisonous, right? T-O-X-I-C, toxic sugar chains. I know. Parents are always saying sugar's bad for you. How can sugar be toxic? But these little things are made up of glucose and other little sugars strung together in such a way that it's really, really much worse than just eating sugar all the time. So now let's finish up this lesson by talking about two types of infection, two options that the virus has, any virus. So this isn't just about phages. All viruses will follow one of these two paths. One of them is called the lytic path, and the other is called the lysogenic. And this, to me, is really hard to keep track of because they both start with ly, and the words are way too similar, and so it's really hard to remember which is which. It's not hard to understand the difference, but the words are just too similar. So let's look at the lytic cycle first, because that's the one that T4 takes. Some phages will do this, but generally T4 follows this. So let's zoom in on the lytic cycle. And you might guess that this big oval here represents an E. coli bacterial cell. And we're going to put a lot of different things happening inside here. At the end, it bursts open, so I drew it open, but when this cycle starts, it's not open yet. But this one picture is going to represent a lot of different things happening. Now make sure your pencil is sharp. I'm going to take a little sharpening time out. I keep a little thing right here next to me so I can sharpen up real quickly. So the goal of either one of these cycles is the same, and that is for the virus to reproduce. That's it. Make more viruses. Nothing beyond that. So in the lytic cycle, we're going to see many viruses produced. This is going to be a little virus producing factory that is then going to release 100 to 150 new viruses into the world. This is pretty straightforward. Number one, the virus attaches. So in this drawing, to make it faster, you might want to just draw a circle for the capsid and a stick for the tail sheath, and then just kind of go like that. In fact, you could even make a circle, a stick, and just a V. That would be good enough. So the virus attaches, and then number 2, DNA is injected. It's going to scrunch down, so make the head close down like that, and kind of the legs up like that. And DNA is injected. So we're going to draw a little strand of DNA going in. Actually, we could make the bacterial DNA. We're not going to use this space for anything else. So we could actually draw a clump of bacterial DNA. Now, the bacterial DNA, I'm not going to say much about it in this drawing. It'll become very important in the next one, the lysogenic cycle. So we can put bacterial DNA. So then, by a process we're not going to go into in this lesson because it's super complicated, a little messenger molecule called messenger RNA is going to be made. So just draw a line with little tiny lines coming off of it. It's like 1/2 of a ladder. This is how it would look, like this. Just see, it looks like 1/2 of DNA. And then we can point to it here, messenger RNA. So the M stands for messenger. So as the word would suggest, it's a messenger carrying a message. So the message it's going to carry is the information that's contained in the viral DNA. Remember, the DNA has instructions on how to make little parts to make a virus. So this DNA, all it says is this is how to make a T4. So a little messenger molecule is made because the ribosomes in number 4, remember those little factories? We drew the little circles in the E. coli. Okay, so we could make some more down here. These little ribosomes, little manufacturing sites, they can read these instructions. Actually, let me zoom out for just a second and show you a picture of a ribosome. So a ribosome is made of 2 clumpy proteins. We're used to seeing them this way, actually. There's a large unit and a small one, and they come together, they click together around this messenger molecule here. So we're gonna draw a little dot. In fact, we can draw a solid dot here with a little RNA going through it. And here's a nice, very simple picture of a ribosome with mRNA going through it. Now here's a ribosome looking much more like a factory. So here's the messenger RNA go in. This blobby kind of brown thing, that's the ribosome. Here comes the instructions through, and there's a little transfer molecule that's bringing up, there's an amino acid. This one is phenylalanine. There's asparagine and lysine, tryptophan already on there. So it's holding on to this little amino acid, and it's going to come in there, and these 3 little things are going to fit with the next 1, 2, 3 on the messenger RNA. So this A is going to match with this U, and this next A is going to match with this U, and this G is going to match with this C. So if you have this UUC, it's going to automatically have the AAG come over, and it's going to stick right there. And when it does, this will get connected onto here. These 2 will get connected. And so you can see these have already been connected, and so you're going to have this stream of connected amino acids going off, a very, very long chain we call a polypeptide. If we zoom out, it might look like this. Here's the messenger RNA, this red tape running through, and here's the ribosome sitting on it, and actually many ribosomes can grab hold and read it at the same time. So you can see this one is just starting the process, and there's a little protein chain coming out. This one has been reading it longer, it's a little bit longer, and this one is almost done, and the protein chain is very long and almost done. It gets to the end, it stops, the little protein chain is cut off, and then what will happen is the protein, there you can see a very detailed version of it. It doesn't look like a piece of string. You can see all the little atoms, but it'll fold up into a blobby shape. So here in our picture, we can make the ribosome as a little dot. Those two little units have joined together. We can make it look like a dot on the messenger RNA. And then we can make here's a little protein coming off like that. Okay, we'll make another one. Make the RNA align with it's like a very long little comb. You could put a dot ribosome on it. And another little thin string coming off. A little protein. So you get all these little proteins then. And they'll clump up. What will they make? They will make the 289 little clumpy proteins that will assemble together to make the T4. And here's a picture of how the base plate will be made of proteins. Every one of these little, like the red one, the blue one, the yellow one, the light blue one, all of these are individual proteins. So that's a lot of proteins that will go together to make just the little base plate. So you're kind of looking down on it here, right? We don't usually see it from this view, or maybe you're looking up from the bottom. So let's make little parts then. They go together. We'll make some heads, some little base plates, some long sheaths, tail fibers. I keep wanting to say legs, they're not legs. Fibers running around everywhere, miscellaneous parts. And then they will assemble to make the completed virus. However, before they do, critical step here, maybe you've already thought of this. In fact, I should move this over. I should have said something about this in here. Part of the assembly process is, though, to make copies of the DNA, right? Every one of those capsids is going to need a full set of DNA instructions, and so the DNA has to be copied over and over again. And I've got to show you this cool demo I made for you about how this happens. I have a capsid that has not been assembled yet. It would be sitting here, and on the bottom of it, it's going to go like this. There is a little protein sitting there that's like a motor, and so it takes in the DNA, and this little motor winds in and it stuffs the DNA in just seconds. They know like exactly how fast this can go. And it goes stuff, 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 stuff. It just reels it in. I mean, my hand's pushing it, but it would just, imagine me just being able to turn it on and it would just suck the DNA in. And it packs that thing so full. They say if you scaled this up, and you could see this, you made this like really big, it would be as powerful as a car engine. And it packs this stuff in at like 900 psi. Pack, 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 tightly full. So then we won't draw all the 100 to 150 of these little things. And you see I have in the note here, they know that this can take place in 30 minutes. That's really amazing. This whole process can take place in less than an hour. And then when there's enough of them, a signal is given and the cell bursts open, and these little guys escape into the wide world. And what do they want to do? One purpose: go find another E. coli. So now we have the ability to infect up to 150 E. coli. And then if this happens in all 150 of those E. coli, that'd be 150 times 150. So in the course of 2 hours, think how many you've made. So this is a very efficient process for replication. Now let's go over here. One other option that viruses might use, and of course they don't decide anything because they don't have brains. The other option is the lysogenic cycle, where sometimes it's to the advantage of the virus to not make a zillion of itself right away. What it does is its DNA kind of hides for a while. And so let's go through this cycle. Number one, the virus attaches like that. And here, the bacterial DNA is going to play a big role. So we're going to make a circle, but I want to use 2 colors. This is actually very important to make 2 colors. So I'm going to make a green circle. We're going to simplify this. And I know that a lot of bacterial DNA is circular. So we're going to pretend we straightened all the DNA out and we just have a circular thing like that. So that's going to be, that's DNA. And then let's see, let's make a little the picture where the virus is shrunk down, scrunchy, and inserted its DNA. And I'm going to choose red. Let's see if I can zoom in a little bit more there. Okay, so we have the virus injecting its genome. And then next, line over like that. The viral DNA is incorporated into the bacteria. So we're going to draw a circle like that, like a C, and then we're going to finish it up with red. So it's a complete circle, but part of it's red. So I made a little string model of this. If this is our combo DNA, it would look, it's actually a circle. If we straighten it out like this. The blue would be the bacteria's DNA, and then the viruses get spliced in. Try to get it all on the picture there. So here's the viral DNA, and it's actually attached. Now, the interesting thing about DNA is it's the same for any organism. Your DNA is basically the same as a bacteria's. You could take bacteria DNA, you could cut your DNA, you could insert, do this, and then there's little enzyme robots that know how to cut and then reattach. There's one for cutting and there's one for gluing it back together. So little enzyme robots that can do this, and that's what goes on here. In fact, we can even make those little dots for little enzyme guys. We can remember enzyme guys. And then what happens next is that the bacteria decides to divide. That's how bacteria replicate. They just split in half. It's called fission. So we could even write here fission, F-I-S-S-I-O-N. Fission means splitting in half. And so you can see it's going to split. It's kind of in the process, but it's already duplicated its genome. That's like one of the first things that happens, is that it makes an extra copy of its DNA. So what is it going to copy? Everything. The bacteria is completely unaware that there's anything wrong with its genome, because remember, this it looks the same. You can splice that very nicely. And so the bacteria just makes a copy of its genome, divides in half, and so you end up with 2 bacteria that have viral DNA like that. So in a way, the virus is already reproduced. So how efficient is this? Well, what happens here? This one's going to split, and this one's going to split. So we can make 2 for this guy and 2 for this guy. We could make 1, 2, 3, 4. So this guy splits like that. This guy splits like that. And you end up with 4 bacteria that all contain that viral genome. And then what happens again? Now we're not going to draw all these because it gets to be a lot of them, right? 2, 4, 8, 16, 32, 64. It's called geometric production. And we're going to, I'll just draw these little things, etc. We know there will be all these. And so off you go. I'm going to write ETC, etc. Cells multiply many times carrying the viral DNA along with them. But you're like, well, there's no viruses though. So at some point, it can come out. If there's some change in the environment, usually, then this DNA can come back out again, and we can do this and make viruses. So we can go over and start doing the lytic cycle at any point. So why even do this? Well, it turns out this is not really a terribly bad thing. They think that in the case of phages, that this actually benefits the bacteria, because the phage would have information in its DNA that's actually helping the bacteria to survive. So then if the conditions become unfavorable and this information is no longer needed, then it can come out and it can destroy the cell, and it can go off and then find new bacteria who need help from a phage. So this is probably part of the natural ecological balance. This is the way that phages help to regulate the ecological systems like the ocean or the soil or whatever.